Hello, everyone. I'm delighted to speak with you today about one of the most fascinating and often overlooked aspects of microbiology, how bacteria talk to each other. Yes, you heard that right. Bacteria communicate, coordinate, and even make collective decisions, all through a process known as quorum sensing. Today, we'll explore these bacterial conversations and, importantly, the therapeutic promise they hold in tackling persistent infections. So, let's begin by looking at what used to be a very solitary view of microbes. For decades, textbooks painted bacteria as solitary creatures, tiny, isolated cells doing their own thing. But as microbiologists, we now know that couldn't be further from the truth. Imagine this. What if these microbes are more like members of a bustling society, constantly communicating, negotiating, and responding to each other's signals? This is the basis of quorum sensing, a type of cell-to-cell communication that allows bacteria to detect how many of their peers are nearby. Why does this matter? Because certain behaviors, like producing toxins, forming biofilms, or adapting to hostile environments, only make sense when performed as a group. Let me give you a personal insight here. When we ignore this microbial social life, we often miss the full picture of how infections establish and persist. Just like we have different languages across human cultures, bacteria use different molecular languages depending on their type. The words in their vocabulary are called autoinducers. These are small molecules secreted into the environment. As the bacterial population grows, these molecules accumulate. Once they hit a threshold concentration, the quorum, they trigger coordinated gene expression. Gram-negative bacteria typically use AHLs, gram-positive bacteria use small peptides, and intriguingly, there's a potential universal language known as AI2 or autoinducer 2 that many species can understand. So, let's explore each of these languages more closely, starting with AHLs. Gram-negative bacteria communicate primarily using N-acyl homoserine lactones, or AHLs. The classic model is Vibrio fischeri, which uses this mechanism to produce bioluminescence. Yes, glowing bacteria. Here's how it works. Lucy synthesizes AHLs, which diffuse freely in and out of the cell. As the population grows, AHLs build up outside the cell. When they reach a high concentration, they re-enter and bind to Luxar, a receptor protein. This complex then activates gene expression, like a switch flipping on a group behavior. This system isn't just about pretty lights. Pseudomonas aeruginosa, a major human pathogen, uses this same system to regulate virulence factors and biofilm production. As I often tell my graduate students, this system is elegant in its simplicity: a molecule, a receptor, and a cascade of behavior. Gram-positive bacteria prefer peptides over lactones. These peptides are often modified after translation and are actively transported out of the cell. Once outside, they accumulate and bind to a membrane-bound sensor, a histidine kinase. This triggers a phosphorylation cascade, activating a response regulator inside the cell. It's a beautifully intricate system, one that's highly specific due to the variability in peptide sequences. Take Staphylococcus aureus, for example. Its AGR system uses AIPs to regulate toxins and biofilm dispersion. Or Streptococcus pneumoniae, which uses a peptide signal to induce competence, essentially turning on DNA uptake. This is molecular diplomacy at its finest, coded signals that lead to precise and coordinated actions. Now, what if bacteria from different species wanted to coordinate? Enter AI2. Produced by the LuxS gene, AI2 is one of the few molecules believed to act across species lines. Think of it as a microbial Esperanto. However, there's still debate. Some researchers argue it's more of a metabolic byproduct than a true signal. Regardless, the evidence is compelling. AI2 plays a role in multi-species biofilms and gut microbial interactions. Escherichia coli, Salmonella, and Vibrio species all produce it. We're only beginning to understand the implications, but as I like to say, AI2 reminds us that even bacteria can be surprisingly cosmopolitan. Now comes the dark side of quorum sensing, its role in disease. Pathogenic bacteria often wait to express virulence factors until their numbers are sufficient to overwhelm host defenses. Pseudomonas aeruginosa, for instance, doesn't waste energy on toxins until the quorum is met. Then it releases a full arsenal. Staphylococcus aureus uses quorum sensing to regulate hemolysins and toxins that help it evade the immune system. Vibrio cholerae coordinates its toxin and biofilm programs via quorum sensing. It's the microbial equivalent of strength in numbers. And for us, it means a single bacterium might be harmless, but a coordinated population can be deadly. Biofilms are another major problem, and quorum sensing is key to their formation. These are not just clusters of bacteria. They're structured communities encased in a sticky matrix called EPS, extracellular polymeric substance. Quorum sensing controls not just the formation of the matrix, but also the maturation and eventual dispersal of the biofilm. Pseudomonas aeruginosa biofilms are notorious for resisting antibiotics and immune clearance, largely thanks to quorum sensing. And Streptococcus mutans, our old dental nemesis, uses quorum sensing to build those robust plaque biofilms. Biofilms are implicated in infections on catheters, prosthetic joints, and chronic wounds. They're a major clinical challenge, and quorum sensing is the puppet master behind the scenes. So, how do we fight back? Traditional antibiotics kill or inhibit bacteria, but that creates selective pressure and drives resistance. What if, instead, we disarm them? That's the promise of anti-quorum sensing, or AQS. Rather than killing bacteria, AQS agents block communication, silencing virulence and biofilm formation. Some methods involve degrading the signal molecules, like lactinases that break down AHLs. Others block the receptors. Furanones from marine algae mimic the signal but act as decoys. We can also inhibit the synthesis enzymes, cutting off the language at its source. This is a hot area of research, and while clinical applications are still emerging, the potential is immense. That said, it's not a silver bullet. There are serious challenges. First, delivery. Can we get AQS agents to the right place in the body? Second, specificity. We don't want to disrupt beneficial microbes. Your commensals might use similar signals. Third, redundancy. Bacteria often have multiple quorum sensing pathways. If we block one, they might use another. And yes, resistance can still evolve via efflux pumps or receptor mutations. Finally, regulatory pathways are unclear. These are not traditional antibiotics, so how do we classify them? Despite the hurdles, the field is pushing forward, and I believe we're laying the groundwork for a new class of microbial therapeutics. To wrap up, quorum sensing governs some of the most critical bacterial behaviors: virulence, biofilm formation, competence. By understanding it, we gain a powerful lens into microbial strategies. And by disrupting it, we may create smarter, resistance-proof therapies. Looking forward, the future of infection control will be collaborative, combining AQS with antibiotics, immune modulation, and even phage therapy. And as master students, your role is crucial. You're not just learning existing knowledge, you're poised to shape what's next. The next breakthrough may not come from a more potent antibiotic, but from understanding how bacteria whisper, shout, and conspire, and learning how to quiet them. Thank you. Please like, share, and subscribe for more videos.
Bye.